Good afternoon. My name is Ken Burness. I'm the Dean of the College of Biological Sciences, and it's my pleasure to welcome you this afternoon for the first in our Tracy and Ruth Storer Lectureship in the Life Sciences lectures for this academic year. Tracy and Ruth Storer were these wonderful people who gave of their minds and their hearts and their riches to enrich the community, both the campus and the Davis community, and we continue to benefit from the legacy of their giving. Some people in the audience actually knew them. I never had the pleasure of meeting either Tracy or Ruth, but their affiliation with the campus goes way back. Tracy was appointed in 1923 as an assistant professor of zoology and served the campus for the next 50 years. This, in the Department of Zoology, this lectureship actually goes back almost 50 years as well. It's been ongoing, and those of you who have looked through your programs recognize the amazing caliber of the speakers that have participated in this series over the years. It's been a great resource to the campus and an opportunity to do something a little unusual, to bring people to campus not just to give a single seminar, but to spend a couple of days and interact with the campus community, the campus and the community while they're here. Ruth Storer shared her husband's commitment to scholarship. She was the only woman graduate of the university's medical school class of 1913 and was the first woman pediatrician in Yolo County. She remained active in the Davis community until she passed in 1986. So we're very fortunate today to have as our speaker Dr. Mimi Koehl from UC Berkeley, and we look forward to hearing her speak. She's going to be introduced by Dr. Angela Cheer from the math department. And I just want to point out one additional thing about this lectureship, which is how wonderfully it transcends the boundaries. It focuses on all of the life sciences, from the most computational to the most observational and ecological. And so we have had a wonderful series of lectures that transcend those. And that, I think, is illustrated by the fact that Mimi will be introduced by Dr. Angela Cheer from the Department of Mathematics.
It is a pleasure for me to introduce Professor Mimi Koehl. Professor Koehl is a professor in the Department of Integrative Biology at the University of California, Berkeley. She earned her PhD from Duke University in zoology, and after doing postdoc at Friday Harbor, which is associated with the University of Washington, and in England at the University of York, she joined Brown University before going to UC Berkeley. Mimi has garnered an impressive list of honors during her career, including, but not limited to, the Presidential Young Investigator Award, a Guggenheim Fellowship, the MacArthur Genius Award, the Berkeley Award from the American Society of Biomechanics for Outstanding Career Achievement, the Rachel Carson Award from the American Geophysical Union. She is a member of the National Academy of Sciences and the American Academy of Arts and Sciences. She is also the heroine of a children's book called Nature's Machines that is part of the National Academy of Science series, Women Adventures in Science. I have known Mimi for a long time, and my impression is that I've always been incredibly impressed with how she takes a problem, looks at all of the data and research, and is able to hone in on the essential, the thing that makes it clear that is the essence of what the problem is. And she has an incredible ability to do that. She is going to talk to us today about settling down. How do larvae of bottom-dwelling marine organisms land in the right place? It is my pleasure to give you Professor Mimi Koehl.
Thank you very much. I'm delighted to be here and honored to be part of this series. So what I want to talk about are bottom-dwelling marine organisms like these corals or barnacles or crabs or whatever. And organisms like this disperse to new habitats by releasing microscopic larvae that are transported by ambient currents. And where these larvae eventually settle back down on the bottom, it turns out to be a tremendously important ecological issue. Where these larvae settle, whoops, I'm going the wrong way. Here we go. For one thing, determines the dynamics and the genetics of metapopulations of these bottom-dwelling organisms along a coastal area or in the ocean at large. And in addition, where larvae settle back down onto the bottom also has a very important consequences for the structure of benthic marine communities. So a very important mechanistic question to address is how do these larvae recruit to suitable habitats after they've been carried around in the ocean currents? And that's the question that I want to address today. For many different types of animals, it's been shown that smells, that is, dissolved chemical cues released by organisms on the substratum, can affect whether they settle or not. And so, for example, members of the same species or the prey of these organisms might release perfumes into the water that cause the larvae to settle and undergo metamorphosis. The evidence for this has come from laboratory experiments with very many different kinds of species of larvae, but the trouble with lab experiments is that they're done in dishes in still water. And anybody who's been to the ocean knows that in nature, the water is anything but still. The flow is very turbulent and messy, and things get stirred up in the ocean. And if you're in shallow coastal habitats, there's also wave action. So larvae are settling in these sorts of habitats, not in a dish in the lab. So the real question that I want to address today is, can larvae use smells in the water to help them settle in the right place in natural habitats? Where the flow is messy and wavy. And I had several predictions before I started out. This is what I thought. I thought that, for one thing, the cues released by organisms on the bottom were going to be way too diluted by all that messy, turbulent, wavy ambient flow, and so there wouldn't be signals big enough to affect larvae out in nature where everything's getting all mixed up. And even if I was wrong about that, even if there are signals, chemical signals out in the messy real world, I couldn't imagine that a little pipsqueak larva, a few hundred microns long, swimming around weakly with cilia, could do anything about it, even if there were chemical cues that it was responding to. So that's what I predicted. I was expecting to find out that all those experiments in laboratory dishes were not going to be relevant to the real world. So before I go any further, I should share the blame with the people that I worked with. The larval experiments and the fieldwork I'll talk about were done by me and by my collaborator, Mike Hadfield, who is a larval biologist who works at the Koehl Marine Lab at the University of Hawaii. And a lot of folks from our lab participated in the fieldwork. It's always easy to get folks to go out on the reef with you. And I particularly want to mention my technician, Tim Cooper, who helped with a lot of this work. I'm going to talk briefly about some mathematical modeling, and it was done by my student, Jim Strother. And I'll talk a bit about some experiments done in a laboratory flume. And these experiments were done at the Environmental Fluid Mechanics Lab down at Stanford with Matt Reidenbach and Jeff Kossoff. So this was a real collaboration between engineers and biologists of various sorts. The research system we used to address this question was this pretty sea slug, Festilla sabogi. But don't let good looks lead you astray. These are voracious predators, and they eat only one species of coral, Porites compressa. So it's critical that the larvae of Festilla land on reefs where this prey species is abundant, or they're dead meat. And Porites compressa is the most abundant, dominant reef-building coral in Hawaii. So I should see how many of you have been snorkeling in Hawaii. A few of you. And if you've seen branching corals, what you've seen is Porites compressa, probably in reefs that look something like this. This is an orgy, literally, of Festilla. And the product of this orgy are these egg cases. And after about 7 to 8 days, the eggs hatch. When they hatch, what's released are little larvae that look like this. This little fellow is about 200 microns in length, and those Mickey Mouse ears are its swimming organ. They're ciliated, and the organ's called the velum, and that's how they locomote around. After about 3 or 4 days in the plankton, these larvae are competent, and what that means is that they're developmentally far enough along that they're able to undergo metamorphosis if they find themselves in a suitable habitat. So what are the steps for a competent larva of Festilla to recruit onto a reef with Porites coral? So if we think about it, the first step in the process is that the larva has to be transported out of the water column to a suitable habitat on the bottom, and in the case of Festilla, that has to be a reef with Porites corals. Once they land there, they have to settle, and what that means is that they have to attach to the substratum. And after they've attached, they then have to undergo metamorphosis from the larval form into the benthic form, in this case a little slug, and they have to survive in order to recruit to that habitat. So the question I want to focus on is, can dissolved chemical cues, in other words, odors in the water, affect any of these processes in the recruitment of larvae to a reef? Now, we already knew before I started this work that dissolved, a particular metabolite from Porites induces Festilla larvae to undergo metamorphosis. Mike Hadfield had figured that out some years ago. In fact, that's why I wanted to use this as a system to study whether odors could affect where larvae land, is because the odor had already been figured out. So I want to back up and say, can dissolved chemical cues from Porites affect larval transport from the water column to a suitable habitat? So let's focus on that step of the process first. So the question I want to address is, can larvae use smells in the water to help them land in the right place in nature? And there are several things we need to know to address this question. One thing we need to know is, what's the water flow like in the field where these larvae are actually going to be landing on the reef? Another thing that we need to know is, in the kind of water flow that happens in nature, what's the dispersal of dissolved cues coming off of the corals? And then we need to know, what do larvae do in response to dissolved cues? Knowing that they undergo metamorphosis is not going to solve our problem, because that's a process that takes about 24 hours. What do they do behaviorally in the short term if they encounter a cue from the Porites coral? And then what we want to do is put all that together and say, do the responses of larvae to cue from the coral affect where the larvae actually land in ambient water flow? So that's what we want to figure out. So let's start out by thinking about water flow in the field. And the place we did our field work is Kaneohe Bay on the island of Oahu in Hawaii. And this is an aerial view of the bay, and it's subjected to waves that roll in off the ocean and move across coral reefs. And if you look here, you can see that there are a lot of patch reefs all over the bay, which makes it a great system for doing research, because you have lots of replicate reefs on which to carry out your experiments. So that's where we did our work, and the first thing that we did is measure water velocity profiles above and within the coral reefs, replicate reefs in Kaneohe Bay. And one of the things that we did was use acoustic Doppler velocimeters that measure water velocities using the Doppler effect. So they bounce sound off particles carried in the water and use the Doppler effect of the sound bouncing back to figure out how fast the flow is going. And we measured water velocities at different positions in the water column above the reef. And let me just show you a typical recording from a position about 2 centimeters above the tops of the corals on a reef. So this is a plot of water velocity, and you can see 0 is in the middle of the graph here. And if you think about how water moves in waves, if you think about it, you know the water sloshes back and forth. And so what I've plotted here on the positive direction is flow shoreward, and in the negative direction flow back seaward. And what you can see is that the water sloshes back and forth, back and forth. So this is wave-driven flow across these coral reefs, which those of you who've been snorkeling in Hawaii have experienced. And you can see that the signal is messy. It isn't just a simple slosh back and forth. There's a lot of wiggling around in that signal. And we can actually, these are the same data you saw, we can ask the question, how much of the variation in flow velocity is due to wiggles at different frequencies? And this is a plot of frequency and how much of the variation is due to frequencies, slow ones and faster ones. And what we see is there's a strong signal due to the waves. And the other thing that we see is that there's a lot of other higher frequencies at which the velocity is oscillating. And what that tells us is that the flow is turbulent. There's lots of eddies there stirring things around, and we have a measure of how much of that stirring is going on. So that's the sort of data that we get in the field above the coral reef. But you might imagine that if you're trying to measure flow by bouncing sound off things, you can't measure flow inside the reef that way because you can't get your sound down in between the coral branches to measure the flow. So we used a different technique to measure flow inside the reef. We used electromagnetic flow probes, and here's one positioned above the reef, but we also have little miniaturized probes, and here's one right at the surface of the reef, and we could embed other probes like that down in the holes within the reef. You can see there's lots of spaces between the coral branches. So we could embed probes at different depths within the reef and measure the flow there as well. And here's a typical example of what we measured. Again, here's velocity shoreward and seaward plotted as a function of time, and you can see the flow above the reef with the waves going back and forth. And here are velocities 5 centimeters below the top of the reef and 16 centimeters down into the reef, and you can see that there's also flow in the reef itself, but it's much slower than flow above the top of the reef. And let's take a look at some numbers. Let's just think about the peak instantaneous velocities when the waves are going the fastest they're possibly going, and compare the flow at different positions. So here's a diagram. Here's the surface of the reef. Here's the peak wave velocity, almost 0.5 meters per second, up above the reef. It's a little slower near the top of the reef. Down within the reef, it's only 8 centimeters a second, and deeper into the reef, it's only about 1/10 of the free stream flow above the reef. So that's the instantaneous peak velocities. We also measured water velocities vertically, because if you think about what water does in waves, it doesn't just go back and forth. It actually goes like this, around and around. And so if you measure the peak, so the water's sloshing up and down into and out of the reef as well. And at this instant, the peak upward velocity out of the top of the reef was about 12 centimeters per second. It's a little slower down deeper into the reef. So there's a lot of flow going on within the reef. Now, as the water sloshes back and forth, if you, for example, toss orange peels into the water in the ocean and watch what happens, you see they go back and forth, but eventually they kind of slosh their way towards the shore. And so if you were to measure the velocity, the net flow over a longer period of time, what you see is that there's net flow across those reefs from the seaward side to the shoreward side. And we can work out what those net flow velocities are by averaging over a long period of time. And what we see is that the flow through the reef, it's sloshing back and forth, but over a long period of time, it's only traveling about 0.5 centimeter per second towards the shore. Flows up out of the reef, net flow up out of the reef at about 1.5 centimeters per second, which is pretty slow. And then there's net transport of water shoreward at only about 3 centimeters per second. So although the instantaneous velocities in waves are really fast, the net transport across the habitat is slow because it's sloshing back and forth. So that's what the flow is like in these wavy coral reef habitats. So what's the transport of waterborne substances like odors coming off the coral in a reef situation like this, in flow like this? And we quantified this by doing dye release studies, and I won't bore you with the numbers, but a picture is worth 1,000 words. You can release dye into a reef and watch what happens with time. And one of the things that you see is after a few wave cycles, the dye slowly flows up out of the reef. There's this slow net flow up out of the reef. And then the dye spreads along the surface of the reef as the water flows by. And because the water is sloshing back and forth, it gets smeared around back and forth in both directions from where it came up out of the reef. So you can see that there's a lot of dye hugging the surface of the reef, but not so much up higher in the water column. So what I've told you is what the flow pattern is like over convex coral heads like this and over flat reef surfaces that look like this. But if you look at a reef, you see that the topography is often much more complex than that. So we wanted to map how water velocities varied with the topography of a reef. So to do that, we laid transects across our reefs, and at 1 meter interval, we measured the net vertical water flow coming out of the reef or going into the reef. And we did that for 6 different reefs, and this summarizes what we found. So I'm going to show you net vertical water velocity. Up is positive, down is negative for different types of reef surfaces. And so up is flow out of the reef, down is flow into the reef. And if we look at those transects across 6 reefs, what we find is that on convex and flat coral surfaces, the net flow out of the reef is slow. It's about 1 centimeter per second up out of the reef. In contrast, if we look at the seaward edge of reefs and measure the net flow, what we find is that there's slow net flow into the reef at the seaward edge of the reef. And if we look at depressions, rather than the convex surfaces, if we look at the belly buttons, the concave surfaces on the reef, what we find is that there's also net flow down into the reef. So the reef is really breathing. There's flow into and out of the reef, depending on local topography. So to summarize, the flow over these coral reefs, the habitat is subjected to very rapid, wave-driven, turbulent flow, which results in very slow net transport shoreward across the reefs. And there's even slower flow through the holes in the coral reef. There's slow net flow of the water that's been flowing in the reef up out of the reef at, that's a boo-boo, that should be convex, at flat and convex surfaces. And there's slow net flow down into the reef at depressions. So there's this predictable circulation pattern of water on coral reefs. So that's what flow is like in the field. How does that kind of flow affect the dispersal of cues released by the Porites coral? So remember, there's very slow flow through the reef. So if these corals are releasing perfumes into the water, we'd expect them to accumulate in the water that's flowing through the reef. And so one of the things we did was go out and slurp up water samples from within the reef, bring it back to the lab, and ask larvae whether it contained the cue or not by asking them whether they would undergo metamorphosis. And what we found is water collected in the reef indeed does induce metamorphosis. So in other words, in the field, water in the reef contains cue in concentrations strong enough to affect the larvae. And we also found that water collected right above the surface of the reef, where you saw the dye so brightly, that water also contains enough cue to stimulate the larvae to undergo metamorphosis. To us, the substances dispersing from the reef look like a diffuse cloud. But what we need to think about if we want to then move on to see how a larva might respond to a cue distributed like this, is we have to say, what is the Q distribution like on the scale of the larva? And remember, the larva is only 200 microns long. So this is how it looks to us. How does it look to this little guy? And so we need to take a much closer look on the scale of a larva at this distribution of odor. And we did that in the laboratory in a wave flume. A flume is just a big tank in which you can make the water move around. And so what I did was take my turbulence and wave data down to the environmental fluid mechanics lab at Stanford, and they set up their big flumes to mimic the field situation that I had measured. And so here's their flume, and what we did is we built a coral reef in their flume using skeletons of Porites. And so this is a side view, and this is a sort of end-on view of our fake coral reef. And then what we did is we wanted to see how dissolved substances released by the corals would be dispersed. And so what we did is use a fluorescent dye that would disperse in the same way as the odor would to visualize how dissolved substances were being released. And that dye was painted onto the corals in a way so that it would slowly dissolve and leach off the corals, just like metabolites would leak out of a living coral. So that's our analog for the odor. And when we do that, it would still look like a big cloud. What we need to do is see it on the scale of a larva that's just a few hundred microns long. And so we did that by making an optical slice through that cloud of dye using a sheet of laser light. So we had this little thin sheet of laser light that would only illuminate one little section of that big cloud of odor so that we could see it on the scale that a larva would encounter it. And that technique is called planar laser-induced fluorescence. So if I lapse into saying PLIF, and you don't know what I'm talking about, I'm just referring to illuminating this fluorescent dye with a sheet of laser light. And here's a frame of a video that we made using this technique. And what you see here are the corals. You can see individual branches. And what you see here is this plume of dye that is leaching out of the corals. And the brighter it is, the higher the concentration. So you can calibrate each pixel of your picture to get the exact concentration of this analog for the odor. And one of the things to notice is that when you make a little slice through it like we did, it isn't a cloud at all. You can see that it's made up of filaments of very high concentration swirling around in odor-free water, which is what looks black. So it doesn't stink where it's black, and it does stink where it's got some color. And so here's the Q, that is the dye leaching off the coral, and here's a larva. And so if you think about what this odor plume looks like on the scale of a larva, you can see this guy is sitting in the middle of the cloud, but he's not in any odor. And if he were to swim across here, he would encounter some odor and then be in no odor again. So the world for that larva is going to be quite different from the way we would encounter this plume of odor. So that's how odors are dispersed over a coral reef. What behavioral responses would a competent larva show to encountering stripes of odor like that, on-off odor? As it swims along, it's going to go into and out of these filaments of high concentration odor. So let's think about what a larva would encounter. Here's a filament of odor, of Q, and here swims the larva, and it goes into and out of the odor filaments. So do larvae respond very rapidly to brief encounters with Q from the coral? And so let's think about what a larva would see. Here's a side view of a larva. Here's its shell. Here's its foot. It has a sticky foot that's a lot like the foot of a slug or a snail. And here's its little ciliated swimming organ. And If the larvae were swimming along, we've timed how quickly they swim. It's about 0.17 centimeters a second. So it's swimming this way. And what that means is that the water flow relative to it is in the opposite direction at that same velocity. So it's just like if you're riding your bicycle this way, it feels like the wind is blowing this way. So we can take advantage of that, and we can have our larvae in bondage. We can tether a larva by its shell so it can't go anywhere, and we can put it in a little flow tank so that we can make the water flow past the larva at the speed that it swims. So even though it's tethered, it thinks it's swimming. It's like it's on a treadmill. So it's happily swimming along, thinking it's doing just fine, even though we've got it tethered. And the advantage is if we tether it, we can watch it through a microscope and see what it does when it encounters odor filaments. And so again, here's its velum expanded and its cilia are beating in clean seawater with no odor. Now I'm going to show you a videotape. Here's our larva tethered, and here's its velum, its swimming organ. And the first sequence I'm going to show you, there's going to be a dye filament, but there's no odor in the dye. It's just dye. That's going to be carried in the flow past the larva. So it would be the same as the larva swimming along, swimming right through an odor filament. But no odor, it's just dye. The second one I'm going to show you is dye plus Q from the coral. So this is the Q that the larvae are metamorphose in response to, and we want to know if it's going to show a behavioral response to it. So let me play the video and you can see what happens. So this is just dye, and you can see that the velum is still out, the larva is still swimming. It doesn't care about dye. Now here's the odor, and bloop, do you see what happened? Pulled its velum in while the Q was there, and when the Q left, bloop, comes back out again. So basically what we see is that when larvae encounter odor from the coral, they pull in their turn off their cilia, pull in their swimming organ, and when they get back out of the stink, out comes the swimming organ, and they resume swimming. So they're little on-off machines, pretty simple. So to summarize, when a larva swims into a Q filament, the velum, the swimming organ, is retracted. And when it exits a Q filament, the velum re-expands and the cilia resume beating. So that's a larva in bondage, but what would its trajectory be if it was not tethered like that? And so we looked at that by putting larvae in an aquarium, and we could either put clean, unsmelly water in the aquarium, or we could put Q from the coral in the aquarium, and then we could illuminate just one plane in the aquarium so it doesn't look like a snowstorm, so we just see a few larvae swimming around in it, and make a video record of the trajectories of the larvae. And let me show you the kind of thing that we found. These are the tracks of larvae in clean, unsmelly seawater. So each dot is the position of a larva on successive frames of the video. And you can see that the larvae are swimming every which way. Now compare that with the trajectories of larvae that are in water that has the inducer from the coral. And what you can see is that everybody is sinking in this case. So if the larvae encounter Q, turn off their cilia, they sink. We can do the same sort of experiment with water we collect from the field. And if we collect water up high above the reef, we see that larvae swim every which way. There's not enough Q to affect their behavior. If we collect water right at the surface of the coral reef, everybody sinks just about. And if we collect water from within the reef, the larvae sink. So we know that the concentrations in nature are high enough to affect larval behavior, so I was wrong. And we did this many times to quantify the speeds that the larvae swim. So this is instantaneous velocity, and these are different batches of larvae. And the open squares are the swimming speed when they're in clean water, and the dark squares are the sinking speed when they're in Q-laden water, so that we were able to come up with what the velocities were for larvae swimming and sinking. And I had a a double major, art and biology, taking one of my classes, who did this animation for me that I'll show you to summarize what competent larvae of Fistula sabogi do. In Q-free water, they swim at the velocities we measure, and when they encounter Q, they sink at a velocity we measured. And so here's his animation. So here's the larva swimming along, cilia are beating, and when it encounters a Q, it sinks. It has a little lag time, which we've measured, and when it exits the Q, it resumes swimming. So it's a simple little on-off machine. Not very smart, but it seems to work, and I'll show you how it works. So we figured out these 3 steps in our process. Now we need to put it all together and ask, do the responses that we've measured to Q actually affect where a larva lands when it's out in the waves crashing around? And, you know, does this behavior make any difference or not? And we addressed that with a mathematical model that my student Jim Strother did. So we had a behavioral algorithm, which is in clean water swim and in smelly water sink, and there's a threshold concentration to get them to sink. And what we did is we put larvae with this behavioral algorithm into the videos that we took in the flume of how odors are dispersed off of a coral reef. And so let me tell you how the model worked. It was an individual-based model for any of you who are into ecological modeling. For each larva, we had it sink if the Q concentration was greater than some threshold. We had it swim if the Q concentration was less than that threshold. And at each time step, the instantaneous velocity of the larva would be its swimming or sinking velocity, and we would determine that by looking at the pixel the larva was in. And we would say, how bright is that pixel, and are you above threshold? If you are, sink. If you're not, swim. Plus, the larva is this little guy being carried in the ambient flow. So we also had an instantaneous local velocity of what the wave was doing, and we had measured that in the flume. So we knew how fast the water was moving at that height above the reef at that instant. And then there was a lot of mess. Remember, there was all those velocity fluctuations due to the turbulence, so we would give it a little kick due to the turbulence as well. And so we'd sum up these velocities and move the larva to a new position. We'd advance the frame 1 frame, and then the larva would say, okay, what pixel am I in now? And what do I do now? And what's the velocity? And so on. And so we could move a larva from 1 frame to the next through our videotape. And let me show you an example of what we got. So I'm going to show you a video made by Matt Radenbach and calculations done by Jim. With the larva sinking in Q and swimming in Q-free water. And the larva is going to start out here. And I want you to watch initially, it's going to be in a lot of water with not much Q, and it's going to go up. And then it's going to encounter a lot of Q filaments and start moving down. So here's a calculated trajectory of a larva following that behavioral algorithm that I just explained to you and carried in the flow. And there he lands on the reef. So one of the things we can do is follow an individual larva and calculate the temporal pattern of Q encounters that larva has. And here's an example. This is Q concentration encountered by a larva as a function of time. So we've just followed it along its trajectory. And what you see is it encounters Q, then no Q. Goes through another filament, no Q, Q, no Q. So what we see is that these little microscopic organisms in the ocean are not encountering a fuzzy, gradual cloud of concentration that's increasing gradually as you get closer to the reef. What they're encountering is on, off, on, off, on, off, because there's so little relative to these filaments. So this is not the way people have modeled how odors are dispersed and how organisms search for a source of an odor. It's usually modeled as a diffusing cloud. But for these little tiny guys, it's on, off, on, off, on, off. And what I've just showed you is what a larva would encounter up at this sort of position relative to the reef. As it gets closer to the reef, you see it has more frequent encounters with Q, so it's going to spend more time sinking. And when it gets very close to the reef, it's almost always above threshold and will be sinking most of the time. So the pattern of on-off encounters changes as you near the source of the odor. So what we did is we didn't just calculate the trajectory of one larva. What we did is place thousands of larvae at random positions in the water column and ran our model and calculated the rate of transport of larvae to the reef following this simple on-off behavioral algorithm. And this is a summary of what we learned by doing this. So let me march you through this. What I've plotted here is what we call transport improvement. And what it is the rate of transport of larvae into the reef for some set of parameters that we're investigating, divided by the rate of transport of larvae to the reef if the larvae didn't respond to the cue at all. They just kept swimming all the time. And also if there was no turbulence stirring up the larvae and carrying them, mixing them around and, in the water column. So basically, the higher this number, the greater the rate of larval transport into the reef. We've plotted it at different Q concentration thresholds. That is, the concentration of Q required to make the cilia turn off. So if we have a low value here, it means the larvae are very sensitive to low concentrations of Q. And at the opposite end here, it takes a really whopping big concentration to make them sink. And in fact, at this end, they're not responding to Q at all. And we also plotted it for different levels of turbulence. So at this end, there's no turbulence. And at this end, it's much more turbulent than we actually encounter on the reef. And what did we learn when we did this? One thing we learned is that the more turbulent the flow out in nature, the greater the rate of transport of larvae to the reef. And that's simply because they're being stirred around. And so some of them are just being mixed and carried down into the reef through no fault of their own. The other thing that we found is as larvae become more and more sensitive to Q, if they're more and more likely to turn off their cilia and sink, the rate at which they're transported into the reef increases. And if you look at this yellow stick here, these are the parameter values that actually occur in nature. This is the threshold concentration for Festula sabogi larvae, and this is the level of turbulence we measure out on the reef. And what we find is that the transport of larvae into the reef is enhanced about 30% over what it would be if they didn't sink when they encountered Q. So it tells us that their behavioral response to Q actually affects where they land. So in turbulent wavy flow, our model predicts that sinking in Q enhances transport rates into the reef by about 30%. And so a model is good for clearing up how you think, but it's nice to test a model. And so we needed to try and test this in the field. And one of the things that the model did was assume that larvae sink into the reef, are retained in the water within the reef. And so we need, once they got down to the reef, we assumed they weren't mixed back up out of the reef. So we needed to test that assumption, and we did that in the field by releasing mimics of larvae. If you've ever tried to rear larvae, you realize you can't rear enough larvae to do a field release and hope to find any of them. So we used particles that sink at the same rate as larvae. We released them upstream of the reef and asked whether or not they were retained in the water within the reef like we had assumed. So the basic experiment was here's the bottom of the bay, here's the reef, here's the net water flow direction towards the shore. We would release dye so we could see where the water moved and larval mimic particles throughout the water column upstream of the reef, and then sample the water above and within the reef at different positions across the reef. And here are all the people out there with their pumps sampling. And that was great fun because every 2 minutes I could say, now, and all these people would bow down with their pumps and take water samples. It was a great feeling of power. And I'm noticing that the time is going by, so I won't bore you with the data. I'll just cut to the chase. So ignore all this stuff that I'm going through because I don't want to talk for too long. What we learned when we did that is that after the dye, that is the water, had moved through the reef, the larval mimics were retained in the water within the reef. So that assumption actually was true. Okay, the model made some predictions, too, that we can test in the field. One of the things that the model predicted was the spatial distribution of larval settlement across the reef. And we predicted that more larvae should land on the seaward part of the reef than the shoreward part of the reef. And one field test that we did was to study recruitment of Fastilla on the reef to see whether that pattern held. So this is a map of Kaneohe Bay, and these are the various reefs where we did a 3-year recruitment study. And one of the things we needed to know is which parts of each of these reefs are seaward or shoreward. And fortunately, there's been a lot of oceanographic work done in Kaneohe Bay, and so there are both data and models of water transport in the bay. So we could focus in on the regions where we worked and see which parts of the reef were seaward and shoreward for each reef where we did recruitment studies. Now there's another problem, which is that new recruits are 200 microns long and the same color as the coral, so it's not like going out and seeing how many barnacles settled on a rock. It's really difficult to see the larvae. So what we had to do was each month go out and collect bits of coral from the seaward and shoreward parts of a reef, bring them back to the lab, and keep them separated in different tanks and keep them in running seawater that we had filtered to 5 microns so no new larvae could get in, and wait for all the babies who had settled to grow big enough for us to see. And after 2 weeks, the slugs are about 6 millimeters long, and we can count them, so we could count the number of Fastilla per kilogram of coral who had recruited. And here's what we found. This is the recruits per kilogram of coral plotted as a function of collection date. And one thing you can see is recruitment is very patchy in time. Sometimes big pulses of recruitment, other times not much. And it's not seasonal. We've got summer and winter bursts of high recruitment. And if we compare seaward and shoreward, so this is again number of recruits, seaward and shoreward on the reef, what we see is, as the model predicted, more larvae recruit to the seaward part of the reef. However, recruitment could be due to landing, which is what the model predicts, or it could be due to attachments once they landed, or it could be due to survival. So to test the landing issue, which is what the model predicted, we needed to ask, where do larval mimics, which aren't going to survive or make decisions about attachments, first contact the reef? And we did that by basically putting flypaper on the reef to which our larval mimics would stick. So here's some flypaper at the tip of a coral branch. Here's some flypaper down within the reef. And we could, again, release larval mimics upstream of the reef and ask what happens to them. And here's the recruitment of larval mimics at different positions on the reef. This is the fore-reef, the mid-reef, and the back reef. And this is the results for 9 different experiments on different reefs. And what we found is more larval mimics also encountered surfaces on the seaward part of the reef as predicted by the model. So both our recruitment study and the larval mimic study showed that that prediction held true. The data I'm showing you are for positions low in the reef. At the branch tips, we found a similar pattern, but much lower particle encounters at the tips of the branches. So what this tells us is that larvae or particles carried in the water are more likely to bump into surfaces deep in the reef rather than at the branch tips. And deep within the reef, there's not living coral tissue. There's dead coral skeleton covered with coral and algae. So just file that away. And also we know that deep within the reef, there's high Q concentration. So what we've learned for this step in the process, can dissolved chemical cues affect larval transport to suitable habitat? Both our model and experimental results suggest yes. I'll briefly tell you a little bit about attachment. Once larvae contact a surface, can dissolved chemical cues affect whether they attach or not? And so to settle, a larva has to stick to a surface, and we wanted to know, can Fastilla larvae adhere to non-Porites surfaces, such as coral and algae, that they would encounter down in the reef, if there's odor around? And so does dissolved Q affect which types of surfaces larvae can stick to, how quickly they can stick, and how tightly they can stick? And we address that question by cutting pieces of living coral tissue or living coral and algae off of the reef and mounting them on slides, which we then put in the sea table and cultured to make sure the tissues would be alive. So after a week, if they were still alive, we'd use them. Here's a side view of that slide, so you can see that they're very flat. Here's this little chip of coral and algae seen from the side. And then what we would do is expose larvae to surfaces like this in a dish with Q from the coral, and then take these slabs at different times after exposure to the odor and measure how tightly the larvae were stuck. Now, how do we measure how tightly larvae are stuck? We put them in this contraption, which is a funny kind of flow tank that produces known shear stresses on the surfaces that we put in there. So we would mount our surface with the larvae sitting on it in that tank and subject them to a range of shear stresses that might blow them off. And then we'd videotape them through a microscope. So we'd watch them, you'd watch the larvae and you'd crank up the shear stress and they'd blow away. And so we could note what shear stress it took to blow them away. And here's what we found. This is the shear stress necessary to dislodge larvae as a function of how long the larva was exposed to the odor. And these are glass slides with nothing biological on them at all. And you can see that the larvae stick pretty well. They stick as well as a newly settled barnacle to these surfaces. So if there's Q in the water, they stick to non-Porites surfaces. If there's no Q in the water, they don't stick. So I'm not even showing you any data because no larvae ever stuck. This is the data for living Porites compressa corals, and you can see that they also stick to living coral surfaces. And this is coral and algae, which they would encounter down within the reef. When they're exposed to Q, the larvae stick pretty well. So if there's dissolved Q in the water, larvae can stick to glass, to coral and algae, and to living Porites tissue. The shear stress to dislodge them is pretty substantial. Like I mentioned, it's a lot like when barnacles first settle, similar stresses. And it takes about an hour and a half of exposure to Q for larvae to develop their maximum stickiness to surfaces. So so we know how tightly larvae can stick, but how does flow through a reef affect where larvae can hang on to the reef to settle? And I'm on the home stretch here. I did start late, but I'm almost done. So the way that we dealt with that was back in the flume at Stanford, we used something called laser Doppler anemometry and what's important to measure water velocities. And what's important about that technique is you can use a laser to measure water flow within a few hundred microns of surfaces at different positions in the reef in a way that you could never do in the field. So we measured water velocities at the height of Fastilla larvae, 200 microns, and we did it at tips of branches on the top of the reef and then on surfaces down within the reef. And if we measure water velocity, here it is 200 microns from the tip of a coral branch on the top of the reef. Here it is 8 centimeters below the top of the reef, and the flow is slower, which is not surprising. But does that matter to a larva? And if we think about a larva, here's a little Festilla larva crawling along a surface, and we modeled it as a sphere. I think that's a pretty good model for these guys. Sometimes we laugh at modeling things as spheres. So we could calculate the net force due to water velocity encountered by this little guy based on our measurements. And here's the force we calculated on a larva at the top of the reef, and then 8 centimeters below the top of the reef. And again, you can see the forces are lower down within the reef. But does that matter to a larva? So to address that, we have to compare these forces with how tightly larvae can stick. And we used that information to calculate the probability that a larva would attach as a function of distance below the top of the reef. And each of these lines is assuming a different amount of time for a larva to make its initial attachment to the reef. So brief times and longer times. And what we found is that larvae are very unlikely to attach to the top of the reef. Their probability of attachment is very, very low, whereas their probability of attachment in the interstices down within the reef is much, much greater. So can dissolved chemical cues affect settlement? Indeed, it can. And so to summarize what we've learned so far, if we ask what's water flow in the field like where larvae are trying to settle, we see that reefs are exposed to waves and that the flow is very turbulent. But as the water is sloshing back and forth, the net flow through and across the reef is very slow. If we ask how are odors released by corals dispersed in water flow like that, what we see is that on the scale of microscopic organisms like larvae, the odor distribution is filamentous. It's not a diffuse cloud. And so microscopic organisms like larvae encounter odors as on-off, on-off, on-off. And if we ask what responses do competent larvae show to this kind of on-off pattern of dissolved Q, we find that they sink in Q above a threshold concentration and swim when they're in clean water. And that sinking enhances their transport into the reef. And we also found that the odor in the water makes them sticky, and sticky enough to have a high probability of actually staying put once they land in the reef. So dissolved chemical cues enhance larval transport to a suitable habitat, induce attachment to that habitat, and induce metamorphosis. So going back to the question I started with, can larvae use smells in the water to help them settle in the right place in nature? What I predicted was wrong. I thought Q would be too diluted by the flow to be useful, and that was wrong. There really is a spatial pattern in odors in the habitat. And I thought larval behavior would be overwhelmed by the flow, and I was wrong about that too. A simple behavior of turning off your cilia when you encounter odor is enough to enhance settlement on the reef. So the answer to this question for Festilla is yes. And what we're doing now is moving on to other systems to see how general this is. So the system we're studying now is not as appealing as a coral reef. This is the fouling community that grows on docks in Pearl Harbor. And there are many different kinds of larvae, worms and barnacles and sea squirts and oysters and bryozoans that show different behaviors and attachment mechanisms. And so we're investigating the generality of this now in this new system. Thank you. Questions? Yeah?
Could you explore other scenarios in your model? For example, if larvae first encounter a plume, they just stop swimming and don't swim again. How much better that strategy would be instead of turning and swimming on and off?
Yeah, so we investigated a number of different larval behaviors, and I'll mention one other before I get to the one you asked. One is, what if you always swim up, which is what newly released larvae that just have hatched out of the egg case do. They're positively phototactic and they swim up. And those larvae are transported away from the reef, and very few go back into their parent reef. So that behavior of very young larvae enhances their transport away from their mother reef to someplace else. We looked at larvae that sink all the time, and in our little model where we always have a reef underneath, more of them settled, but in nature that wouldn't be a good strategy, because if you just encounter one little filament, you might have washed across a coral head and there aren't any more, and you might settle someplace inappropriate. So it would be fun to develop a model where we had a patchy substratum, which we haven't done yet. We've got the whole substratum releasing odor. So that would be something fun to explore in the future. Yeah?
I think maybe you just answered this, but if you had in a model part that was reef, part not, how does that deal with the probability of settling in the wrong spot?
Yeah, so what happens, and I didn't get into this either, but as you, if the substratum is no longer releasing Q, then what happens to those filaments with time as they're mixed around in the turbulence and those filaments diffuse is that the concentration gets lower and lower and lower. And so eventually, if you're downstream, there isn't much odor, so you're not above threshold. So you might expect, if the reef were tiny enough, that you might get a shadow of settlement on the wrong place behind it. But most of the larvae are depleted from the water column before they get across the whole reef, so that, at least in our model, that wasn't a problem. Most of the larvae were gone by the time we got to the back. But in deeper water, that might be an issue. So that'd be fun to look at, too. Yeah?
Do these larvae adapt with time? Is this sort of set point for their behavior constant, or does it change as a function of their history?
So we explored that in our little treadmill for larvae, and they're pretty dumb on-off machines. One of the things I didn't mention, which is a parameter we varied in our model, is that they don't respond to every filament. They respond to some proportion of them, something like 70% of them. And so we varied that as a parameter in the model, and it has just the effect you would imagine. If they respond to fewer of the filaments, then the settlement is less. But we didn't see a time trend in them learning about what they were smelling or adapting to it and ignoring it. Yeah?
What's the effect of the tidal patterns on the flows?
At our particular reefs, there was no, the tidal, regardless of time in the tidal cycle, the flow was always the same direction across the reef because of the configuration of the bay. The waves drove the flow across the reef, and then there were channels at the end where the flow would go back out. So we never had, the upstream end of the reef never became the downstream end at our particular sites. But you would expect in habitats where the tidal current would change that I would predict that you'd have a lot of larval settlement on both edges of the reef and less in the middle. The other thing that happens with the tide is that the water depth changes. And the other thing that happens during the day is that in the afternoons, the wind typically picks up, so the waves get bigger. So there's a lot of temporal variation in the flow patterns that we saw. So what I was telling you was sort of typical for the afternoons when the water was deeper and the wind was higher. So some of the time it's less flow than what I showed. Yeah?
Do larval, they normally settle during the day or during the night, or is there?
We don't know. When you do laboratory experiments for our metamorphosis assay of how strong the Q is, they'll settle anytime. But I don't know in nature when they settle, because our only way to know whether they settled is to bring them in and wait 2 weeks. So I can't answer that. But in the lab, they'll settle any old time. I don't know if that varies temporally. It's just a metabolic waste product from the coral, according to Mike. Do you know Valerie Paul? Yeah, she's analyzing it right now, so we're sitting on tenterhooks waiting for her to tell us what it is, so I don't know what it is yet.
Do they respond that way to amino acids or
No.
nothing?
No, it's just, and so when we were making our inducer for these various experiments, what you do is you just put the coral in some seawater overnight, and then it's full of odor, and then, yeah. Yeah?
Do they exhibit any positive or negative responses to conspecifics at all?
No, not that we can tell. Yeah, not that we can tell, yeah.
Okay, and how specific is the response of these larvae to the Fistula stupid or whatever?
Say that again?
How specific, I'm trying to think of a name for the mysterious substance.
The perfume?
Yeah, the
Yeah.
variety stupid or the
Yeah.
Fistula stupid.
So we've taken the other species of coral on the reef and made coral water from them in the same way, and the larvae don't respond. They don't behaviorally respond, and they don't metamorphose. So it seems to be, it's specific for the genus Parietes, and there's only one species of Parietes in Hawaii, but there are other genera of Parietes in the Pacific that will cause Fistula to respond as well. So it's at that level. Yeah?
Is the sticking, is that one way? I mean, like if you have glass, they stick to glass. Do they, if the Q disappears again, do they release or do they stay on the surface?
They have to be, I'll back up. We know that they have to be exposed to Q for at least 4 hours to undergo metamorphosis. And if they've been exposed to Q for 4 hours, then it's too late. They will undergo metamorphosis. But we haven't yet done the experiments where we put them in Q, get them to stick, and then put them in unsmelly water to see if they'll unstick. So I can't answer that yet, so I don't know. But I do know after 4 hours, they're committed to metamorphosis. Yeah?
In all this turbulent flow, or maybe with temperature changes or dissolved gas, do you expect variations in water density, and so do they compensate buoyancy to sink all the time, when they're not swimming?
They sink because they have a shell, so they're really dense. The temperature of the water is pretty constant during the day when we've gone out there with our thermometers, so that doesn't change very much. And I think if there was a big rainstorm so that you got fresh water and lower salinity, they would just die. So that's all I know about that. Questions?
So the increased settlement in the forereef that your model predicts, is that simply due to the higher density of larvae in the forereef? In other words, does the model take into account the decrease in larval density as the larvae pass through the reef?
That's right. So what that is due to is depletion of water from the water column as it passes across the reef. And these reefs are in very shallow water, so that we only have a meter or less of water above the reef. So we might get a different result if we modeled a deep water column with a lot of mixing of larvae in vertically. So in our model, all the larvae are being carried in the water that's coming horizontally onto the reef. And so it is due to depletion.
It's very pretty for a pilot.
Oh, yeah.